scientist at MD Anderson. He's federally funded. And I think that, I've seen him in the context of doing a lot for organized neurosurgery. He works with CNS tirelessly. I remember when Chris Getch was the president in DC. I mean, we would get emails late at night. He was working really hard on that program, and he was a real, right now you're treasurer or secretary of the CNS. So thank you so much for taking the time, and without much further ado, Dr. Ganesh Rao.
Can you hear me okay? I've got this mic on. Dr. Nanda, thank you so much for the invitation, the kind invitation. It's really a pleasure to be here. We've worked together for so many years now, and I love talking to you about stuff outside of neurosurgery. We both enjoyed the musical Hamilton in New York recently, and I spent some time with your residents today. They're fantastic, very bright, so it's a pleasure to meet with them. So I'm going to spend the next 45 minutes or so talking about what my laboratory is based on, which is really trying to understand the process of malignant progression in glioma. So I'm going to take you through kind of our current understanding of the biology of low-grade glioma, describe the current management strategies. We'll describe the process of malignant transformation in the context of what's called apoptotic suppression. So I've sort of focused on the suppression of apoptosis, or the disabling of apoptotic mechanisms, as a way for these tumors to become more malignant. We're going to take a bit of a digression through the utility of murine models and studying brain tumors, because they're quite informative and helpful, and that's what I spend a lot of my time doing, is kind of understanding the biology through mice. And then no brain tumor talk would be complete without the discussion of immunology these days. So I'm going to sort of talk about that, touch on that at the end of the talk, and how we've sort of investigated the role of the immune system in facilitating malignant progression. So it's not a common tumor. Low-grade gliomas represent about 16,000 new cases per year in the United States. Most of those are astrocytomas. About a third of those are oligodendrogliomas. There are so-called mixed tumors, although that diagnosis is falling out of favor. You contrast it with about 200,000 new breast cancer cases or lung cancer cases a year. This is not a major player. In fact, at MD Anderson, primary brain tumors don't even crack the top 10 as far as new diagnoses go at our place. But for neurosurgeons and neuro-oncologists, this is a very vexing problem. And so we see a lot of these because they come to our attention. Historically, low-grade gliomas have been diagnosed by histologic criteria, and I'll discuss those in more detail. Typically by cellular pleomorphism and hypercellularity. And again, that's included astrocytomas, oligodendrogliomas, and mixed tumors. And patients may have a lengthy, somewhat indolent course initially, but, and there's always a but with these low-grade gliomas. It used to be historically that we would just observe these cases, and we'd even call them benign. But what I wanted to understand, and what has become clear over time, is that these tumors will almost inevitably transform to something more aggressive, and when that happens, survival declines precipitously. So again, they're generally graded by histologic criteria. Here we contrast what we see with high-grade gliomas and low-grade gliomas. With the high-grade tumors, you'll have increased mitotic activity, microvascular proliferation, necrosis. You don't have any of this with the low-grade tumors. And this has historically been what's been used to diagnose these tumors. But the molecular criteria may be more accurate than histologic criteria, and this is becoming now part of our integrated diagnoses for glioma. So for example, now we know about things like IDH1 and IDH2. IDH1 may in fact be an initiating event in gliomagenesis, an early event. We know now that R132H is the most common IDH1 mutation that can be identified by immunohistochemistry. So in our pathology lab, our pathologists actually do a quick IHC on all our glioma specimens and tell us as part of the diagnosis if it's IDH1 mutated or wild type. Astrocytic tumors can now be differentiated by the ATRX mutation. This is also an immunohistochemistry test that can be performed in the pathology lab. That's alpha thalassemia mental retardation syndrome X-linked. It's almost mutually exclusive with 1p19q loss, which defines oligodendrogliomas. And for patients who have a loss of ATRX, that's associated with a more favorable prognosis. So it is some useful information that is now becoming available. What has been known for a long time is the fact that oligodendroglial tumors are defined by 1p19q loss, so chromosomal losses which are identified by FISH, and now the genes on those chromosomes have been described just a couple of years ago. These are both functioning as tumor suppressor genes, and so it's likely in time we'll find some use for how to restore their function. Now, how do we manage these low-grade gliomas? Well, for a long time, as I mentioned, observation was kind of the norm. When I was training many years ago now, if somebody showed up with a low-grade glioma, we would say, great, you've got a low-grade glioma, these are benign, we can just observe them, and they would go on their way, and some of them would do very well for many years. We know now, thanks to work by many individuals, that we should probably be intervening in these patients. So things like extent of resection, which have been championed by people like Mitch Berger and Hughes Defoe from France, they've shown that there's almost an incremental benefit to removing these tumors with surgery. And I'll talk about how that can potentially mitigate malignant transformation to a certain extent. And RTOG 9802, which was started many years ago now, that data is now maturing. So the preliminary results were reported in Journal of Clinical Oncology in 2012. And at that time, the data wasn't quite yet mature, and it wasn't clear that there was a benefit to using so-called PCV chemotherapy and radiation. But now the data is matured, and I'll show you a little bit of that data now as it's come out. The bottom line is that we're seeing an improvement in median survival for those patients that get a combination of radiotherapy up front followed by PCV. So for those of you that remember the observation days, we would argue that these lesions can be stable for some time. Hughes Defoe has shown quite convincingly, I think, that these lesions almost, always grow. If you heard him talk, he's a champion for kind of pointing out that these lesions are not stable. And we all fall into this pattern of maybe looking at the last scan that we got on a patient and just comparing those two. But if you go back far enough, you'll notice that there's growth. And if you look at the x-axis here, you can see that we're talking about years, potentially decades of time it takes for these things to grow. But when they do transform to more aggressive tumor, the survival drops precipitously. The extent of resection data is also quite compelling. So this is work that was published from the Norwegian group in 2012. Most of us are familiar with this now. This is, Mitch Berger talks about this as being level 1 evidence, kind of prospective randomized data. It really isn't. What it more represents is two different hospital systems that had different philosophies. So a hospital that really preferred resection versus one that preferred biopsy. And what gets really emphasized here is that the survival data is quite different between these two groups. So we don't know much about the extent of resection here. We don't know, how much they were able to get out. We just know they had a philosophy of being more aggressive with these tumors, and their patients did better. What gets a little bit lost in this data, which I think is also equally compelling, is the fact that the malignant transformation rate, so going from a grade 2 tumor to a grade 3 or grade 4 tumor, is significantly lower in the patients who had resection. So again, not only can we help these people live longer, but we can also potentially mitigate their transformation to a more aggressive phenotype. And here's the RTOG 9802 data. So as I mentioned, this has not yet been published, but little bits and pieces of it are being presented at ASCO and SNO and those sorts of meetings. And the data is quite compelling now that PCV followed by radiation provides a dramatic survival benefit for these patients. So in fact, we, at our place, our neuro-oncologists have adopted this for subtotally resected low-grade gliomas. And I should mention that in this trial, patients who got a gross total resection went on observation. So again, if we can get it all out, we're probably better off watching those people, and you're going to give them a benefit, survival benefit, and also hopefully prevent that tumor from turning into something far worse. So what does this mean? What is malignant transformation, right? So I've mentioned to you that given enough time, these tumors almost inevitably degenerate to a more malignant phenotype. They pick up other hits, and it was known for a long time, even before any of this molecular stuff was known, that there were demographic risk factors. And some of these are probably surrogates or proxies for some of the molecular stuff. So older age. That probably meant people with wild-type tumors, right? Because IDH1 mutant tumors have a predilection for patients who are younger than that age. Tumor size, KPS, eloquent tumor location, absence of seizures, these were all risk factors for tumors becoming more aggressive. We now know things like IDH1 wild type, the lack of a co-deleted genotype, intact ATRX, and an astrocytic phenotype as opposed to an oligodendroglial one are all risk factors for becoming more aggressive. So let's show some examples here of what this means. Now, I emphasize these cases because they don't play by the rules. Even though I've told you there's all these factors that we think are favorable, there's obviously other issues going on. That's what I like to investigate in my lab. So here's a 39-year-old man who has a medial left frontal non-enhancing tumor. Turns out to be IDH1 mutant, low-grade astrocytoma. and no malignant features at all in this. We got a gross total resection, and I thought, this is a couple years ago now, actually 4 years ago now, that this guy was going to do pretty well. He's 39, he meets some other criteria, he could be around for another decade or so. We got a gross total resection. 2 years later, his scan looks like this. And he has recurred terribly. He's gone through chemotherapy, radiation, none of it's worked, and he was dead just a few weeks after this scan. Here's another patient. I actually showed the residents this case earlier today. This is a patient who had a IDH wild-type low-grade astrocytoma. So wild-type, now although he's younger, he's got kind of a bad phenotype here. And for patients who maybe don't fit all the criteria, you sometimes have to sequence these patients. This guy had his tumor sequence just to verify that he didn't have one of the what we call non-canonical IDH mutations. And he doesn't. He's purely wild-type. But here he is 7 years later with a slight recurrence that we took out, and that's still a low-grade tumor. So, some of these people don't play by the rules, and what I want to try to figure out is why that happens. Now, we know now that the heterogeneity of glioblastoma, for example, is really overwhelming. So, Bradley Bernstein at Harvard published this last year showing that single-cell sequence in the tumor, so the same tumor, intratumoral heterogeneity, is really remarkable. The same tumor can have multiple different EGFR mutations, for example, where it was thought that those would be uniform throughout the tumor historically. And then sort of roughly the same time, the Tavare group showed that, again, within the same tumor, you could have multiple clones that exhibit different types of genetic heterogeneity. So this makes the problem of understanding the biology much more complicated. More recently, we just published in Cell, I was fortunate to be part of this group that looked at the diffuse subsets of diffuse glioma. And what we basically showed, and I won't get into this too much, is when we look at diffuse glioma, that includes kind of low-grade tumors. We're now identifying multiple subtypes of glioma. So there's the IDH mutant and wild type, and you can see several subtypes that fall out from that. This follows the pattern of what we know about glioblastoma. As long as 10 years ago, it was shown that there are multiple subtypes that all have different clinical outcomes and genetic outcomes. So these are things like proneural, classical, mesenchymal, and neural. Of these, the proneural and mesenchymal seem to have the most disparate gene expression patterns and clinical behavior, and we've sort of settled on those as the different groups that we focus on. Here's work from Ken Aldape's when he was at MD Anderson showing the difference in gene expression between proneural and mesenchymal. If we look at the work that Roel Verhoek did more recently, we see the different subtypes as far as gene expression patterns go. And why that's relevant is that the proneural subtype, again, associated with markers that are associated with neurogenesis, relative to the mesenchymal tumors, it has better overall survival, so clinically it's a better actor. And this is a subtype that seems to be predominant among what are called, again, the low-grade gliomas that are secondary glioblastomas that transform from these low-grade gliomas. The mesenchymal tumors, again, appear to have markers of tissues of mesenchymal origin. Relative to proneural tumors, they have a poorer overall survival. You see kind of the worst histologic features, things like angiogenesis and necrosis, and these seem to correlate more with the primary glioblastomas, the ones that sort of form de novo. Although there's emerging evidence from Eric Holland's group showing that probably all glioblastomas start out as proneural, but this is a continuum, and they pick up genetic hits and become more aggressive and become the mesenchymal phenotype. So this is just the clinical data showing that proneural gliomas do better than mesenchymal proliferative tumors. This is MD Anderson data and UCSF data. So multiple factors contribute to malignant transformation of low-grade gliomas. I've just mentioned a few here. Pro-proliferative cell signaling to promote tumor cell growth. What I've liked to focus on is anti-apoptotic signaling to inhibit tumor cell death, keeping the tumor cell alive so it continues to pick up genetic hits. The induction of angiogenesis, immunosuppression, and emphasizing the fact that heterogeneity of glioblastoma makes treatment very difficult once that transformation has happened. So these are sort of the hallmarks of cancer. I'm sure many of you are familiar with this. What I've focused on is cell signaling programs that sustain proliferative signaling, programs that resist cell death, i.e. suppress apoptosis, genetic programs that induce angiogenesis, again, a hallmark of glioblastoma, and then how does a tumor avoid immune destruction? So we've turned to the Cancer Genome Atlas to study this. It's something that is online and available, but unless you have a bioinformaticist, it's very difficult to navigate, so I've been lucky to partner with a couple of these individuals who are very helpful, and you can download the data. And the reason that's helpful is you can then start to do some differential expression of the RNA data from these data sets. So here we looked at differential expression of anti-apoptotic genes, which are listed here, between low-grade tumors and high-grade tumors. And what we found was several of these were differentially expressed, and we sort of ranked these things. I'm going to focus on what's called BRCA5 or survivin first, because it's instructive on how our lab sort of puts things together. So BRCA5 is not a simple gene. It actually has 3 dominant isoforms. It's a member of the inhibitors of apoptosis gene family, which I'll talk about in more detail. It has 3 variants. 1 and 2 are anti-apoptotic, which are of interest to us, and variant 3 appears to be pro-apoptotic. Previous studies had shown that survivin has a negative prognostic effect on high-grade glioma. So patients with high expression of this did worse than those who didn't. And that predates any of the TCGA data. But what's not known is what the effect of individual isoforms were on survival. So we analyzed expression data from the TCGA to determine the impact of these anti-apoptotic isoforms on survival and progression-free survival. And just by way of background, the inhibitors of apoptosis gene family is basically a family of genes. There's 8 of these BRCA genes, and they basically bind caspases responsible for apoptosis. So they inhibit programmed cell death. This is, again, survivin or BRCA5 is the most well described. More than a decade ago, Arnab Chakravarty showed that those tumors that had high expression of this, those patients did much poorer survival than patients who did not have it. Interestingly, it's not seen in normal adult brain. It's only seen in tumor cells, which made it a very appealing therapeutic target for a very long time. In fact, a lot of tumor vaccines, for example, were used survivin as an antigen to try to target the tumor. So when we look at the low-grade data, this is from TCGA, we see that for patients who have what we call overexpression survivin, so the expression data is normalized, we pick out the patients that have elevated expression relative to the normalized mean. And we see those patients do very poorly compared to those what we call non-expressors. In this case, these patients have a median survival of about 17 months, which is pretty close to what a GBM has, as opposed to the patients who are called non-expressors, and they were surviving almost 100 months, and so doing much better, what we would expect for patients with low-grade glioma. Now here's kind of how our process worked for this. So first we look at the effect of survivin expression in the provisional low-grade glioma data set. I just showed you that. We downloaded that data. Then we looked at it in all GBMs to see how it correlated not only in the low-grade phenotype but also in the high-grade phenotype. Now I mentioned to you that there's multiple isoforms, so I wanted to know how those different isoforms behave, the different variants. And then finally, we overexpress the individual variants in vivo to determine their effect on malignant progression using a murine model. So I'm going to take a bit of a digression here to focus on animal models because they're quite instructive, and this is, again, what I spend a lot of my time doing. So why do we use these models for studying brain tumors? Now, let's focus on a couple of very common models first. Xenograph models or allograft models. Both of these systems rely on cell lines that have been passaged for time immemorial. We can implant them in the brain or in the flank. They're easy to use that way, and you basically can grow a tumor very easily. They generally involve implantation of cell lines in nude mice. These are mice that lack an immune system, so they don't really quite represent the normal tumor microenvironment. A lot of cell lines are available that are either passed from your colleague down the hall, or you can buy them from ATCC, for example. But they don't really grow like a normal human glioblastoma does. So this is, U87 is considered a glioblastoma cell line, but I would argue that looks nothing like a glioblastoma in a human being. It's a well-circumscribed tumor that grows radially. It seems to push the brain away. It really doesn't invade. And U87 is actually derived from a 44-year-old man with a GBM almost 50 years ago. I mean, nobody knows what this tumor was at that time. That predates many of the WHO grading schemes and certainly the genetic characteristics. But there are some advantages to these systems. They are easy to generate. They form tumors reliably and predictably. I can get a tumor with U87 basically 100% of the time when I inject the cell lines into a nude mouse. We can manipulate these cell lines to determine oncogenic effects. And they're frequently used for preclinical testing in novel therapeutics. So a lot of mice with U87 have been used to test new therapies. They seem to always respond when you read the papers, but that almost never correlates with a clinical benefit when we take those drugs to the clinic. There are a lot of disadvantages. As you can tell, I'm not a huge fan of these models, these xenograph models. They really don't recapitulate the key features of human tumors. They lack microvascular proliferation. They lack necrosis. kind of grow these balls of tumor lacking an invasive phenotype. And because you're putting an already malignant cell line into the brain, you can't really study the acquisition of additional genetic abnormalities as the tumor kind of grows in situ. For my purposes, you really can't use them to study malignant progression because there really are no low-grade glioma models. There are no cell lines out there that you can use to grow in mice. And again, there's a lack of a functional immune system. There are allograft models, so as opposed to taking a human cell line like U87, there's GL261, which gets used a lot and is touted as a immunocompetent model. This was developed by putting a carcinogenic agent into a mouse and then identifying tumor cells and then culturing those tumor cells. And you basically do the same thing. You take those and you implant them back into black 6 mice. They're immunocompetent, so you can study the immune system, but I would argue the brain tumor itself is not quite the same as a human tumor. And they grow radially just like U87, but appear a little more invasive than U87. There are glioma stem cell-based models. So these are models that we use a lot at MD Anderson. These are actually derived from patients, and the cells are grown as neurospheres in stem cell media. They can be profiled, so we have publications out now showing how they represent the proneural or mesenchymal phenotypes. They do grow invasively. They do exhibit microvascular proliferation and necrosis. They still need to be grown in NOD-SCID mice, these non-obese diabetic immune-compromised mice. So you're still lacking an immune system there. So the flip side of this are genetically engineered mice, which is what my lab focuses on. So that actually, that's kind of a term that describes a variety of murine models. Traditionally refers to models that recapitulate the genetic changes that give rise to human disease. It allows for the study of molecular causality for tumor initiation, progression, therapeutic response, and histology. These are sort of what we call knock-in or knockout mice. They can have conditional mutations or inducible mutations, which are important because sometimes you want to restrict those mutations to certain tissues in the body. It also describes other models that rely on gene expression that may be vector-mediated, and that's what I focus on, and I'll spend some slides talking about that. And the important thing is these generally give rise to spontaneous brain tumors. You're not taking an already malignant cell line and putting it into a mouse. You're seeing how the tumor arises from the natural environment and see how that it can pick up additional genetic hits to become more malignant. They do have several theoretical advantages. They allow, again, for more accurate modeling of activation of oncogene and inactivation of tumor suppressor genes, since those can be knocked out. You can precisely control the cancer gene activation in mice spatially and temporally, and it, to a certain extent, recapitulates the random nature of sporadic human tumors. You're modeling tumor from the tissue of origin, and again, transgenic mice, by and large, have intact immune systems, so you can study how the normal blood vessel system gets co-opted by the tumor to facilitate its growth. There are some disadvantages. There are some basic physiological and genetic differences between humans and mice. They're very expensive, and when you want to add drug testing, that can add even more expense. And there's a significant time investment. This has been cut down a little bit by things like speed congenics and CRISPR-Cas systems, but in general, it can take up to a year just to make one mouse. And then tumor formation is often variable. Your phenotype may not represent what you expect it to be. And there's really actually no data yet to show that these genetically engineered mice are any better at predicting response than the cheap and easy xenographs. So we use the RCAS-TVA model. This is a model that's developed by Eric Holland, who's a neurosurgeon at Fred Hutchinson now. And this is a model that essentially uses postnatal gene transfer. So unlike some of these genetically engineered mice where you mutate a gene from the beginning and the mouse grows up with that mutation, we're doing this postnatally. And we can do it in a cell type specific manner. So we can restrict the cells that are being affected by the gene of interest. Basically, it uses a chicken virus, it's an avian leukosis virus, it's a retrovirus, and a transgenic mouse line that has a receptor for that chicken virus. And we can modify that chicken virus to express a gene of interest, and it will only go to the cells that are expressing the receptor for that chicken virus. Expression of that receptor can be driven by tissue-specific promoters. So now there are several of these models out there that can model all kinds of different cancers. So we've settled on the one where the receptor for the virus is restricted to expression by the Nestin-positive cells. These are neural progenitors. And here you can model gliomas and actually medulloblastomas, and I'll talk about that a little bit. But there's several other tissue-restricted promoters that express the receptor to model other types of cancer. And several models have been described using different types of receptors or different types of tissues to model different types of tumors. So as a graphical representation here, just to make it clear, basically the receptor for the virus, which is known as TVA, is expressed only from nestin-positive cells. That restricts the incorporation of a gene of interest into glioneuronal progenitors, which are essentially consistent with the cell of origin for gliomas. Here's a representation of the vector. So again, it's a retrovirus. We incorporate a gene of interest here. These are retroviral genes that are spliced out once they get taken up into a mammalian cell. And here's the receptor for the virus. And then this is incorporated throughout the host genome there. And the LTRs, the long terminal repeats, act as promoters. So everywhere this goes in the cell, the gene is being cranked out. And that's how we can restrict it to a particular cell type. So we've used the PDGFB dependent model glioma to study genes implicated in malignant progression. So PDGFB, or PDGF I should say, has been implicated in gliomagenesis and described as an early event in tumor formation. So we've known for a long time that aberrant activation of or activity of the platelet-derived growth factor pathway can initiate gliomas. And again, we use mice that express the receptor to take up the RCAS vector, and we inject newborn mice with the virus, and the gene of interest is expressed in nestin-positive cells. And the reason we do newborn mice is that's when the population of nestin-positive cells is at its highest. You're getting your highest likelihood of incorporating this gene into the genome. And again, these mice are immunocompetent, which is a departure from xenographs. So there are some advantages to this system. Again, spatiotemporal gene transfer into somatic cells in vivo. So what do I mean by that? So if I inject PDGF into the frontal lobes of these newborn mice, I will get a glioma in the frontal lobes. Conversely, when I started getting into this, I was working with Dan Foltz at the University of Utah. We were modeling medulloblastomas. And if I take sonic hedgehog and inject it into the posterior fossa, into the cerebellum, I'll get a medulloblastoma. So there I'm targeting the granule neuron precursor of the posterior fossa. So you can see how versatile this is with respect to being able to target particular anatomy in the brain. We can do simultaneous or sequential transfer of genes, so I can either inject a bunch of RCAS vectors together or do them in sequence. I can correlate that, or I can combine these mice with gain of function or loss of function models. So for example, I've crossed the NTVA mice with P53 knockouts, and that way I can see what P53 is doing in the context of PDGF overexpression. We can use marker genes to trace gene expression to see what happens to that gene over time as the tumors grow. In highly penetrant models where we have tumors forming in over 90+% of the mice, we can use them for preclinical testing, and I'll show you some data of that. And again, we can exploit the immunocompetence. So tumors arise after a transformative event in normal cells. They form in the natural stroma. They really do recapitulate the histologic features of human tumors. Here I'm showing you a high-grade glioma, so you see pseudopalisating necrosis, microvascular proliferation. And then unlike that ball of tumor you saw with U87, you see a very invasive tumor that's growing through the parenchyma. There are disadvantages, of course. No model is perfect. We are limited to about a 2.5 kilobase insert into the vector. If you don't get anything, negative results are sometimes difficult to reconcile. You have to prove the gene was expressed in order to verify that there was an effect. Tumor penetrance is incomplete in some circumstances, so if you only get 25% of the mice forming tumors, that may be due to low infection efficiency, and then you can't really use that system for preclinical testing. Gene expression level may be influenced by the integration site into the host genome, so depending on where those genes land or those vectors land when they get incorporated, that can affect expression. And then there is the theoretical possibility, that when you randomly integrate into the host genome, you can influence the expression of host genes, what we call insertional mutagenesis, for example. Although that has not been a tremendous problem for us. So I'm going to take it back to modeling, how we use that model to model the inhibitors of apoptosis in these mice using the RCAS TVA system. So I showed you the data about low-grade glioma with BRCA5 or survivin, and showed how there was a significant survival disadvantage to those tumors or those patients who had a high expression of that. Now in GBM, there was no difference when we looked at those patients. We see increased expression in almost half the cases, but there's no survival disadvantage compared to low grades. When we dig deeper and look at the different subtypes, so this is the proneural gliomas, we see separation of the curve. And we see a significant difference in progression-free survival for the proneural tumors. And that's important because, again, these are the tumors that are more likely to represent the secondary glioblastomas, the ones that are going from low grade to high grade. In the mesenchymal tumors, the so-called, probably de novo GBMs, again, we don't see that. So that was interesting to us because, again, we wanted to study this in the context of malignant progression. And it turns out that survivin is actually preferentially expressed in proneural glioblastoma. So not only does it confer a survival disadvantage to the proneural tumors, but it also turns out that they're more likely to be overexpressors. So almost 2/3 of the cases, proneural cases, were overexpressors of survivin compared to just about 1/3 of the mesenchymal cases. So then, as I mentioned earlier, there are two anti-apoptotic variants, and so we needed to see how those affected, or how those were expressed in these GBM cases. So it turns out most of them are variant 1 and only about 10% are variant 2. When we look at the survival data, the patients that have variant 2 were living much shorter than patients that had variant 1, or even the non-expressors. And when we look at the proneural group specifically, this is all GBMs. We look at proneural group specifically, we see the same thing. The patients with variant 2 were surviving shorter than the patients who were non-expressors or variant 1 expressors. So then we want to know, how can we verify this effect? And we turn to the mice for that. So as I mentioned, PDGF seems to be an early event in gliomagenesis. When we express PDGF alone in NTVA mice, we can induce low and high-grade gliomas. The predominant phenotype with PDGF activation alone is the low-grade phenotype. So about 3/4 of the tumors end up being low-grade, about 1/4 are high-grade. We created ARCAS vectors to ectopically express human survivin variants 1 and 2. The vectors were expressed in PDGF and independently in NTVA mice because we didn't know if survivin independently was enough to cause a tumor. Nobody really ever looked at that. Then we follow the mice for 90 days or until they develop symptoms. And the reason we chose 90 days is by that point, if the mice are going to get a tumor, they should have one. And we want to humanely euthanize them and not let them go too long unnecessarily. So what we found actually was that, in fact, variant 2 seems to be the subtype that promotes malignant progression. So with variant 1 combined with PDGF, we see, with both variants, we see a very high tumor formation rate, over 90%. With variant 1, most of them are low grades, 3/4. That's essentially the same as what we see with PDGF alone. So whatever this is doing, it's not driving malignant progression. Conversely, with variant 2, we're seeing over 80% of the mice developing tumors that are high grade. And if we look at the histology, we see, again, a bland hypercellular tumor consistent with a low-grade glioma with variant 1. With variant 2, we're seeing microvascular proliferation and pseudopalissatic necrosis. And here we're just demonstrating expression of the variants that we put in there. We also, in addition to seeing a higher grade phenotype, we're seeing a survival disadvantage for the mice that are co-expressing PDGF and variant 2 compared to variant 1. Variant 1 and variant 2 independently without PDGF are insufficient to form tumors. So clearly they're not an initiating event, but an additional hit that drives malignant progression. So I thought, maybe the variant 2 is has a more potent anti-apoptotic effect than variant 1, and so we quantified that by staining for cleaved caspase 3, and it turns out there is no difference, that they're both supplying the same degree of apoptotic suppression. But we did notice that there was more microvascular proliferation in the variant 2 tumors, and so we quantified that with CD31 expression and also looked at microvessel density and microvessel counts and found a higher level of vasculature within those tumors. And I didn't know what to make of that until we found this paper that was published in Blood by a leukemia group showing that, in fact, variant 2 is very highly expressed in vascular endothelial cells, and its localization there is distinct from the common survivin isoform, which is survivin variant 1. So it turns out that this variant 2 is actually critical to endothelial cell formation. And they also similarly did not find an increase in apoptosis relative to variant 1 in their studies, and no changes in proliferation. So it turns out that this is likely conferring some effect in the tumor to recruit new vasculature. So to summarize this part of the talk, BRCA5 or survivin seems to be predominantly observed in proneural tumors relative to mesenchymal ones, where it has a poorer progression-free survival in this subtype. Variant 2 is more relevant to tumor progression because we see poor tumor-free survival in mice relative to variant 1, increased high-grade tumor formation, and increased microvascular proliferation, which is consistent with what other investigators have found. So variant 2 may be a more relevant therapeutic target than the common isoform. The problem is it's only expressed in about 10% of glioblastomas, so that makes it less appealing. Now, to take you back to this heat map, we were looking at other inhibitors of apoptosis, And as I mentioned, we looked at BRCA5 first, but it turns out BRCA3 is also equally, if not more so, interesting. When we look at the data compared to other IAPs, we find that its fold is a log scale. Its fold change in GBMs relative to low grades is significantly higher. And its survival of the patients who are overexpressors of BRCA3 in low grades is significantly shorter than the non-expressors. Now, BRCA2 also demonstrates that. But what's interesting is that the TCGA data gets updated frequently. This is actually data from several months ago. When I looked recently, this survival disadvantage for BRCA2 actually went away, and that's because they keep adding more cases. And so you've got to be careful when you look at the data. It's not static. It actually gets updated, and they add more clinical data. But for BRCA3, this survival disadvantage has remained constant. For both progression-free survival and overall survival for BRCA3 expressors in high-grade glioma, there's a survival disadvantage. It's statistically significant. And we have access to patients who have had a diagnosis of low-grade glioma, and they come back to Anderson many years later with a malignant transformation. And so we looked at a panel of these tumors. So here's a patient who had a low-grade glioma, actually an oligo, and then who came back 7 years later and has significant expression of BRCA3 in their now high-grade tumor. And when we looked at these matched samples, we got a panel of 8 of them that we've looked at. For example, low-grade glioma 1 and high-grade glioma 1, these are all paired up. You see that there's a significant increase in BRCA3 positive cells in the high-grade tumor relative to the low-grade tumor. This is correlation, not causation. So to study this in more detail, we went back to the mice. And here's expression of BRCA3 in NTVA mice. So in fact, we do see for PDGF alone about a 75% low-grade tumor phenotype. But in the mice that are expressing PDGF and BRCA3, a significant increase in the high-grade phenotype. And there's actually, not surprisingly, a survival disadvantage in those mice as well. This is showing you kind of the difference in those, what we see phenotypically in those mice. So again, in the PDGF-induced glioma, it's a very bland hypercellular tumor, but in the PDGF plus BRCA3-induced glioma, we see microvascular proliferation, a much more invasive phenotype than PDGF alone. We see a significant decrease in apoptosis. So in the PDGF tumors, you see these apoptotic bodies scattered throughout the tumor. They're less prevalent in the tumors induced with PDGF and BRCA3. And when you uncouple that suppression of apoptosis from tumor cell proliferation, you see much more significant mitotic activity, and that's just depicted here. So I'm going to switch gears a little bit and kind of finish up the talk with a discussion of how we've investigated immunosuppression and malignant progression. So one of my colleagues at Anderson, Amy Heimberger, who's also a neurosurgeon but also an immunologist, came to us to see whether or not we could correlate malignant progression with anti-apoptotic signaling. And we had noticed on our own that there was a significant increase in immune cells in these tumors. What's the link here? Well, anti-apoptotic genes can drive malignant progression. We've shown that. But we also showed they induce immunosuppression. And that's work we did with Amy several years back, because you see an influx of immunosuppressive macrophages and an influx of regulatory T cells, which are the T cells that kind of shut down or put a brake on the immune system. And we've shown that you can reverse that now to improve survival in mice and mitigate transformation in more malignant tumors. So we showed a few years ago now that infiltrating macrophages are present in high-grade tumors in NTVA mice. So the mice that had higher-grade tumors had a much higher appearance of these infiltrating macrophages throughout them. And mice that died early on in that 90-day course also had a higher incidence of infiltrating macrophages. So we know that these immunosuppressive macrophages are a key driver of immunosuppression. Tumor-associated macrophages are known to be a major constituent of the tumor microenvironment. In fact, if you look at a GBM, somewhere between 30 to 50% of the tumor mass are macrophages. It's made up of microglia, which are the resident macrophages in the brain, and macrophages from the peripheral circulation. So they're recruited to the microenvironment, and we think now that the tumor is sort of co-opting those to keep the CD8 cells, for example, the anti-tumor CD8 T cells from attacking the tumor. The reality is that the macrophage biology is extremely complicated. They can be tumor suppressive or supportive. You'll hear people talk about this M2 skew. That's a characterization of the macrophages that indicates that they are actually immunosuppressive, whereas the M1 phenotype appears to be tumor suppressive as opposed to tumor supportive. And a variety of markers have been used to characterize these immunosuppressive macrophages. And it's very confusing. I mean, you'll see different reports. This just came out a few months ago, kind of describing the various ways to characterize these macrophages. We've tried to keep things simple. So again, exploiting the immunocompetence of NTVA mice, we find that they possess many of the same immune cells observed in humans and have characterized them as such. So CD11b, CD163, CD204, and IBA1, which is a very nice stain for immunosuppressive macrophages. And again, you can see how much of the tumor is taken up by these macrophages. What we wanted to know is how does the microenvironment contribute to malignant progression? So here are mice. Again, you can see the infiltration of macrophages. This is the IBA1 stain throughout the tumor mass, and it actually nicely tracks invasive tumor cells on the H&E. And these are matched human tumors. So these are human low-grade tumors. These are 3 patients who then progress to high-grade tumors, and you can see the increased macrophage infiltration in the high-grade tumors. I put this slide in here just to kind of differentiate, again, the utility of the genetically engineered models to GL261, for example, which gets touted as, again, as an immunocompetent model. What we noticed was that, and this is a manuscript we're working on for something else, but it points to again, how the macrophages really do seem to correlate with the areas of high-grade tumor and track where those tumor cells are infiltrating to. But in GL261, what you notice, so here's the tumor, again, growing kind of as a nice discrete ball as opposed to an invasive tumor. But the macrophages seem to line up on the periphery. They don't really invade. So this is periphery here, and this is center, and you don't see them invade, as opposed to the NTVA mice, where they seem to go both not only in the periphery, but also into the central tumor. So what drives that? So obviously these macrophages don't get there by themselves. There's something pulling them in. And so I'm going to just quickly talk about STAT3, which is a molecule that I worked on, again, with Amy Heimberger, which is a key molecular hub of a variety of pathologic pathways that are activated in glioma. And not to belabor the point, but one of the most important things about this is that it induces M2 macrophages, and that's been shown by many investigators. And so we modeled, and the STAT3 is called a master regulator because it influences a variety of cellular programs, including apoptosis, including immunosuppression, angiogenesis. And so when we model STAT3 in our mice, we obviously get a very malignant phenotype. We also looked back at the TCGA data and found that STAT3 co-occurs with IBA1 and correlates with poor survival. So in tumors where you see a high level of STAT3 and IBA1, that macrophage marker, you see a survival disadvantage. We've inhibited STAT3 with a small molecule inhibitor called WP1066 in the mice and have shown that there's a survival advantage to those mice that are treated, and there's a commensurate decrease in the percentage of macrophages in those treated mice. This is a drug that's actually about to make its way into clinical trials at our place, I think later this year. And it also modulates the tumor microenvironment and immune response. So not only do you get a survival advantage when you use that drug, but you're also seeing a decrease in angiogenesis with the treated mice. That's compared to TMZ. So temozolomide, for example, is a control in untreated mice. And you see a decrease in the F480 positive macrophages in the treated mice. We've also taken an alternative strategy to inhibiting STAT3 with microRNA to epigenetically silence it, and we see a survival advantage here. This is work we just published in JNCI a couple years ago with a decrease in the macrophages in the tumor microenvironment. So to summarize here, if you leave with anything, hopefully you'll appreciate the heterogeneity that GBM possesses that makes alternative therapeutic strategies appealing. And one of those strategies is certainly targeting the microenvironment of the tumor. And that's something that we've seen in melanoma, non-small cell lung cancer, kidney cancer now with targeting the immune cells with CTLA-4 monoclonal antibodies and PD-1 or PD-L1, which has really led to some long-term cures. It's possible that we might see that in glioblastoma. Those trials are just being started now. And again, targeting the tumor microenvironment in glioblastoma is appealing. It's one of the most immunosuppressive cancers in human beings. And what I'm interested in is knowing whether or not we can mitigate that malignant progression by targeting the tumor microenvironment, because we know now that those cells are being drawn in to the microenvironment and potentially facilitating this transformation. It's difficult to know when, if you're going to target the microenvironment in low-grade glioma, when do you do it? Do you do it early on in the process? Do you wait for a certain amount of time? So those are questions that I think still need to be answered. We are continuing to investigate factors that contribute to the progression from low to high-grade glioma. Using these available knowledge bases, they're very powerful, and data is being added to them all the time. We're focusing on transcription factors like STAT3 to see how they facilitate not only immunosuppression, but also apoptotic suppression. And we're continuing further studies on the effect of the immune system on tumor progression. So we actually have crossed our mice with CD8 T-cell knockouts and conditional macrophage knockouts in our colony to investigate the relevance of these cells to tumor progression. And we want to investigate the impact of apoptotic suppression, specifically on facilitating immunosuppression in the tumor microenvironment. Because we can stop that, and there are many agents that are out now, particularly leukemia and that sort of thing, that may have application to glioma. So obviously, this work was done by a number of people. In my lab, Latha Khatri, Tiffany Doucette, and Yihui Yang, Amy Heimberger's group, Fred Lang's group is a neurosurgeon, one of my colleagues at Anderson, everybody in pathology, my biostatistician colleague, Arvind Rao. Still keep in touch with my mentor back at Utah, Dan Fultz, has been incredibly helpful to me over the years, and I've been fortunate to have a variety of funding sources. So with that, I will gladly take any questions you may have.
Any questions?
Do you have a tumor bank? Do you have a tumor bank, like if people send their tumors from across the country, or do you just work with your local referrals?
That's a good question. So we've been fortunate that we have collected these tumors over time. It's very difficult to get matched samples, which is what I'm interested in. And so fortunately, our tumor bank goes back to like 1994. And some of these patients, it's like a 10 to 15 year time span between low grade and high grade. So we've collected about 250 of these patients just at Anderson. Now, some of those I've gotten from elsewhere. They were eventually treated at Anderson for their high grade tumors, and I've called their referring center to send me the specimen. But we don't have a formal mechanism to collect those tumors. I wish we did. It's a very critical resource that I think a lot of people can benefit from.
I have sort of a philosophical question. Charlie Wilson was from Louisiana.
That's right.
And his dream was to cure glioblastoma in his lifetime. And I guess there's a GBM case that Dr. Soon did, and he had a great line. He says, glioblastoma, it's not a surgical disease.
Right.
And do you think in your lifetime you will see it cured? Or, do you think in the next, you think about it, it's not like Gleevec came and CMLs are cured. But it just seems like, Stoop protocol, all that's helped, but it's still such a...
I think what you're going to see are subsets of GBM that are cured. I think there are going to be, and I was telling your residents about it earlier, the IDH story, right? So IDH1, there are IDH1 inhibitors now. They've cured some relapsed AML patients or cholangiocarcinoma patients. Whether or not that biology is the same as it is in glioma, I'm not sure. And the preliminary data for IDH1 inhibitors for high-grade glioma is not good. So the data is emerging now, and it doesn't appear those inhibitors are working. But for low-grade gliomas, there does appear to be some effect. The problem is timing. When do you start giving people these drugs? Because so are they going to take this drug every day for 15 years, hoping they don't progress to a higher-grade tumor? For fully malignant GBM, I do think, I think immunotherapy holds some promise there because it's a very immunosuppressive tumor. I think it's like melanoma, right? 20% of stage 4 melanoma patients are effectively being cured with immunotherapy. So could you see 5 to 10% of that? You could. The TCGA data has shown that there's some novel fusion proteins, just like with Gleevec, right? So BCR-ABL with Gleevec, you cured those patients. Now, it doesn't turn out, in GBM, it turns out that some of these fusion proteins aren't as biologically relevant as they are for leukemia, for CLL. So people have targeted them, and the results preliminarily aren't that impressive. But I think in my lifetime, God, if you're not hopeful in this field, you're doomed. So I think that. I think that
Jimmy Carter's 91 with that checkpoint inhibitor.
He did,
cured. Like they said, you don't need any more follow-up. You're done.
Right. And he had metastatic disease to the brain.
He had metastatic. He had the gamma knife.
A gamma knife and
So what's the role nowadays for interstitial treatment? Is there any role for locally delivered?
You mean for anything?
for GBM? Or
so you mean like gliadel, or?
Yeah.
So we've shied away from it. I mean, the problems, the data is not that compelling. Henry Brem was just at our place a couple months ago, and he's still obviously a big believer in it. And they're advancing that field. I would say that certainly at our place we don't use it. And there have been some other kind of aborted attempts at it, like Gliosite, where you inject, you put a balloon in the resection cavity, and there's some radioactive agent. That company, I think, went under. So
how about catheter-based, the stuff Dr. Lang used to do?
Yeah, so CED, he used to do p53 gene, and that did not work. I think CED, frankly, is one of those things that we've heard about for 20 years, and I've never seen anything result from it, so I'm not sure that's the next step. Some people have touted oncolytic viruses, so Fred has moved on to an engineered adenovirus, which targets a retinoblastoma mutation essentially on the tumor cells, and there's been some preliminary data from that showing some responses. So oncolytic viruses are hot right now, but I think the interstitial treatments are probably still have sort of fallen out of favor.
And then these cases, if you get a new, low grade, a lot of these tumors are quite big, often in eloquent regions. Yeah. So if you get a brand new tumor like that nowadays, what is your how do you start from diagnosis to?
Yeah, so we have sort of drunk the Kool-Aid of being very aggressive. So we will, if we can, we'll do as much surgery as we can. Justin Smith and Mitch Berger showed many years ago that there's an incremental benefit to extent of resection. So with GBM, we sort of have this cutoff of 78%, 80%. If you can't get that much out, you may not be doing much good. But with low grades, I think it's different. I think the more you can get out, you're providing some benefit there. So again, for RTOG 9802, and even at our place, if you get a gross totally resected low grade, let's say it's a tumor that is amenable to that, totally in the frontal lobe or something where you can be very aggressive, we'll observe those patients, but we'll definitely try to take it all out, as opposed to a diffuse one where we'll take some of it out, frankly, mostly for diagnostic reasons, but maybe a little bit of therapeutic benefit. And now our oncologists will go with PCV and radiation, or radiation and PCV. I think that's going to become the standard of care.
So they don't use Timidor for low grades?
No, I mean, they don't feel like for low grades that Timidor has the same effect as PCV. It's certainly better tolerated, but they'll start with PCV.
And I like the point that you made from Jacola's paper that I didn't realize that the incidence of malignant progression in the radicals was much lower.
Much lower.
That's an important point. Yeah. Well, thank you so much.
More or less awake surgery nowadays?
Are we doing more or less?
Yeah, awake surgery.
We're doing more, I think. We have a low threshold for it, and so for my, I think for speech, you have to do it awake. For motor, you could argue that sleep mapping is pretty good, and I think it is, but there's something just more comforting about asking the patient, can you move your arm? You feel that immediate sense of relief that they can move it. So I would say we're probably more likely to do things awake than not.
Yes.
You showed data from RTOG 9802. Was that the long-term update, or was that the?
No, that's still not out yet, as far as I know. I've got some slides from it that I'm not supposed to show anybody.
But I know the early, it showed a difference in progression-free survival, but
not overall.
Right. But the new data supposedly has a benefit in overall survival.
It does, absolutely.
But I haven't seen it.
So I've seen. So Jan Buckner, I think, sent some slides to somebody who sent them to me, and there's clearly a benefit in overall survival. Yeah. So that's again why we're moving. That paper hasn't not come out yet, but
I know, it's been a long time.
Long time.
And that's why I think a lot of places are still relying on PCV, because all those trials, they take a long time to gestate.
Yeah.
Back then, if PCV was the treatment of choice, there's a new trial out that's going to hopefully let us know whether temozolomide and PCV are equivalent, but that's of course not even finished yet, not close yet.
Well, on behalf of the department, I'll clap for you.
Very kind of you.
Thank you so much.